Today we're going to start Unit 7 for Microbiology of Characterization and Classification of Prokaryotes. Now remember, prokaryotic cells are going to be things such as bacteria and archaea. With these types of cells, remember they don't have any membrane-bound organelles except for the ribosome, and they are going to be a single-celled organism. Now with prokaryotic cell morphology, you can have several different types. The first type you could have is going to be referred to as a cocci. That's going to be that singular coccus. That's a spherical shape that you see there in green. Next, you can have the bacilla. Go to this. So here's your cocci. So that's going to be one single circular cell is going to be a coccus. Then we're going to have the bacilli. That is going to be a singular rod-shaped cell. So we have a coccobacillus there. Notice how the ends of it are kind of circular, and then we have the regular bacillus here. Now that coccobacillus is going to be an intermediate between the cocci and bacilli. Next, we have the vibrio. That's going to be what we refer to as a common shape or a comma-shaped bacteria cell shape that we will see sometimes. Up on the right hand side, you will have what's called a spirilla or spirillum. That's this one here. It's often referred to as being a stiff spiral shape. Next, you have your spirocate. That's going to be a flexible spiral shaped organism here. Then you have your phleomorphic, and that's going to be various shapes and sizes. So you'll see all of these different shapes and sizes in there. And then, of course, you see the star shape there. You can also have what's called like a triangular shaped one. You can have more of a rectangle shaped bacteria. So those are all going to be different types and shapes that you can see when looking at these prokaryotic organisms. Now coming in, you can also see how they're going to be kind of put together and clumped together. So when they put them together, let's look at the different cocci up here that's going to be on the left-hand side. You could have what's called a diplococci. That's where you're going to put them together in pairs. So if you see them paired when looking at them under a microscope, this pairing is going to be referred to as a diplococci. Then you can have what's referred to as a tetrad. So looking here at these tetrads, notice that they are cocci, but they're going to be put together in clusters of 4. Next, we're going to look at streptococci. Strep means that it's going to be in a chain. So if you go to the doctor and you have a strep infection, that means your bacteria are going to be in the chain. So if it's a streptococcus bacteria, it is going to be a chain of cocci. Okay, next one that we see here is going to be called a scarinae. That's going to be cuboidal packets. And so you see how it's going to be in a nice cube shape, but it's almost like 2 tetrads on top of each other. And then coming down to the bottom one, notice how it's this nice big clump. That is going to be a cluster of cocci. And so that's going to be referred to as staphylococci. So if you say that you have a staph infection, this is how they're going to appear when they grow that bacteria on a petri dish. You take the bacteria, you make a slide, and when you make your slide of it to look at it, these are going to be how each one of these appear on those microscope slides. Now moving on over to the right-hand side, we're going to look at the rod shape. So these are the bacilli. These first ones with the single bacilli, you could have them in singles, you can have them in pairs, you can have them in chains, or in what's called a palisade. That's going to be a folded type arrangement that you see here. So you could see a staph, that member is going to be chains of these bacilli, or you could have a diplobacilli where you're going to have 2 of those together. I've seen those quite often. And then sometimes you'll even see them making these V-shape formations. So all of that's going to lead to helping you identify what type of organism it is according to how it is arranged in how it arranges its cells together. Next coming in, we're going to talk about reproduction. So with prokaryotes, they're going to go through reproduction where an offspring is going to arise from a single parent, and that offspring is going to inherit genes of that parent. Now there's going to be 3 different types of asexual reproduction that we have in prokaryotes. The first one is going to be binary fission. That's going to be the most common method of asexual reproduction. So here the parental cells are going to divide into two identical daughter cells, and then those parental cells will disappear with the formation of their offspring. Let's go back here and look at the picture a little bit closer. So with this, notice how you have your nucleoid. That's going to be your circular DNA. It is going to be attached at some point to the cell membrane. Sorry, thought I brought my tool up. So it's going to be attached, and that is not drawing. Okay, let's see if we can get this to work one more time. Sorry about that. And so you see it's attached to the cell membrane at a specific point. It's going to double up on its DNA, make a second nucleoid. They're going to be identical genetically. And so it's going to attach itself. The cell is then going to start to elongate itself. It then starts to pinch in right here at these cleavage furrows until eventually it pinches apart completely and makes two identical daughter cells. So you start with the one cell and eventually you end up with two daughter cells. They are going to be genetically identical here. Now with the next type, we refer to this as snapping division. This is going to occur in some gram-positive bacilli. So the parent cell's outer wall is going to tear apart with a snapping movement to create daughter cells. And so you can see that rupture of the outer wall and the hinge there. Okay, next we have budding. This process is going to be where the outgrowth of the parent cell receives copies of genetic material and enlarges, and then it's going to detach. Other modes of reproduction, you could have spores. This is going to be where reproductive cells that develop into clones of original organisms. These are going to occur in actinomycetes and fungi. Next, you have fragmentation, small motile filaments that are going to glide away from the parental strand. This is going to occur in things like cyanobacteria. Next, you have viviparity. This is the production of a live offspring. Offspring are going to emerge from the body of a dead mother cell, and it occurs in epuloposciscum. So next we're going to go into endospores. Looking here, you can see that endospores are going to be seen on the left-hand side in green. And then when you look over on the right-hand side, they're going to be the ones that are in the darker reddish pink color. So with these, they're going to be unique dormant structures that are going to be produced by some bacteria that are going to be a defensive strategy against unfavorable conditions. They're highly resistant to damage caused by physical factors such as heat, pressure, radiation, desiccation, which is drying out, toxic chemicals such as antimicrobial drugs, so it really helps them to survive. When we talk about pressure, think about pressure cooking and pressure canning things to save food. So this is how some of those bacteria can exist and can continue to live later on. So they're going to remain inactive, but potentially viable for long periods of time. They're able to resist stages that because they're barely metabolizing here. So they have very resistant abilities. Sporulation is going to be a process of endospore formation, and it's going to require about 8 to 10 hours when it does this. Endospore formation can occur centrally, subminally, near like one end or the other, or terminally at one end, and they're going to produce gram-positive bacillus and clostridium. Now looking here, we're going to have three domains. We have archaea, which we're going to see here in the middle. I'll get my pencil up. So we have our archaea in the center. We're going to have bacteria, and we're going to have the eukarya. Now with the bacteria and the archaea, these are going to be single-celled organisms. They are going to have no nucleus and no membrane-bound organelles except for ribosomes. When we get into the eukarya, these are going to be the ones that are going to actually have the organelles. They're going to have cell membranes that are going to surround a lot of these different organelles. With archaea and bacteria, they also usually have cell walls. So all domains are going to be based on genetic relatedness to RNA sequencing. So So going into the archaea first, we're going to have three major differences between archaea and bacteria. Cell walls do not contain peptoglycan in the archaea. They do in bacteria. Cell membranes are going to contain lipids with branched hydrocarbon chains. And the AUG codon for methionine, or the start codon basically, is going to be the initial amino acid in the polypeptide chain. Two major groups of archaea are going to be under first we have the extremophiles. The first one is going to be your thermophiles. Now when I talk extremophiles, that means that they're going to live and require extreme conditions such as temperature extremes, pH extremes, or salinity. So with the thermophiles, these are going to be microbes that thrive in temperatures above 45 degrees Celsius, or that's 113 degrees Fahrenheit. Hyperthermophiles are going to thrive in temperatures of 80 degrees Celsius or 176 degrees Fahrenheit. DNA membranes and proteins do not function properly at lower temperatures, so they require these in order for these three things to function properly. These are going to be primarily found in places like hot springs and the deep sea floor hydrothermal vents. Next, we have our halophiles. With the halophiles, these are going to be microbes that thrive in environments with high salt levels, so about 17 to 23% salt concentration. High salt levels are going to be necessary to maintain the cell wall integrity, and and you'll have bacteriorhodopsins, which are going to be purple proteins that harvest light to synthesize ATP. These are going to be found in Halobacterium, primarily found in extremely saline habitats such as the Dead Sea and the Great Salt Lake. Next, you have your methanogens. This is going to be the largest group of archaea. They're responsible for converting CO2, H2, and organic acids into methane gas, which is CH4. They play significant roles in the environment by converting organic waste in ponds, lakes, and oceans, and they're important in industrial areas because they help with sewage treatment. Primarily, these are going to be found in marshes, swamps, mudflats, marine sediment, and gastrointestinal tracts of humans and other animals. Now we're going to go to our second domain, the bacteria. Bacteria are going to be classified according to differences in RNA sequence. So deeply branching and phototropic bacteria, you have your deeply branching first, which are going to be our RNA sequences and growth characteristics, which are similar to bacteria. They are autotrophic. They're found in hot, acidic, anaerobic environments, which means they don't have oxygen. And there's going to be three different representatives of these. You have your aquifix. That's the earliest branch of bacteria. They're chemoautotrophic, which means that they use chemicals for food. They're hyperthermophilic, which means that they like extreme hot conditions. And they are anaerobic, which means they don't require oxygen. They derive their energy in carbon-forming inorganic sources in very hot habitats with little oxygen. So that's what's important about them. The second group is going to be the Deinococcus. That's resistant to heat, pressure, desiccation, which is drying out, and exposure to radiation. And the third type is Thermus. They're hyperthermophiles that are associated with hot springs and deep sea hydrothermal vents. Next, we have our phototropic bacteria. These are photosynthetic lamellae. They have pigments that contain non-membrane bound thylakoids, and that's going to be responsible for absorbing light, usually from the sun. This is going to allow that phototropic bacteria to acquire energy that's going to be needed for anabolism. Autotrophic, so that means that they're able to make their own food, and there's going to be three main groups of these based on pigment sources of electrons for their photosynthesis that they do. So the first type is going to be referred to as cyanobacteria. These are going to be blue-green bacteria, or you might more commonly know them as algae. They are phototropic. They are oxygenic. That means they have the ability to produce molecular oxygen, or O2, which is breathable oxygen, and they reproduce through binary fission. Subspecies are going to be capable of nitrogen fixation. So that reduces the atmospheric nitrogen, which is N2, to NH3, So it takes that nitrogen gas, basically, which was the N2, and makes it into ammonia. That's going to be your NH3. It's going to be important in that it is going to be able to provide nitrogen in usable forms, and it has heterocysts. This is going to be specialized thick walls that are non-photosynthetic cells, which are going to be used to fix those nitrogen. Now, when you look at these underneath the microscope, you can see it on the left-hand corner. I think they're beautiful. They have this definite, distinct shape. So when you look at it, they're usually bright green or sometimes have kind of a bluish green color to them. So they do look amazing. I like to look at these. Next, we have our green bacteria, which you see at the bottom. These use bacteria chlorophylls for photosynthesis. They are anoxygenic, so they do not generate oxygen during photosynthesis, and usually you'll find two different types. You have what's called the non-sulfur, so they derive electrons through carbon dioxide reduction of organic compounds such as carbohydrates and organic acids. And then you have the sulfur ones. These derive electrons from oxidation of hydrogen sulfide to sulfur, and they deposit sulfur outside of the cell. So they're going to be found in anaerobic muds that are going to be rich in hydrogen sulfide at the bottom of ponds and lakes. And our third type is going to be the one all the way on the top to the right. That is your purple bacteria. These are going to be used with bacteria chlorophylls for photosynthesis. They are anoxygenic, so they don't generate oxygen during photosynthesis. And we have the same two different types. We have the non-sulfur and the sulfur. The non-sulfur are going to derive electrons from carbon dioxide reduction in organic compounds such as carbohydrates and organic acids as well. And the sulfur type are going to derive their electrons from oxidation of hydrogen sulfide to sulfur, and they deposit that sulfur within the cell. These are going to be found in anaerobic muds that are rich in hydrogen sulfide at the bottom of ponds and lakes. The next thing we need to discuss is going to be the G+C ratio. This is going to refer to the percentage of guanine to cytosine base pairs found in the genome. So remember, A's with T's, C's with G's. So this is the C's with G's here. So with low G+C gram-positive bacteria, that's going to refer to bacteria with ratios of guanine to cytosine base pairs below 50%. So there's two main types. You have Clostridia, that's anaerobic. Remember, does not require oxygen. They're rod-shaped. They have endospore forming abilities and typically are going to be associated with soil and or water. This will cause things like gangrene, tetanus, botulism, diarrhea, and dental plaque. The second type, we have mycoplasmas. This is a gram-negative. Nucleic acids are going to be studied that indicate more closely related to gram-positive bacteria, though. They're highly pleomorphic, which means that they have variable shapes. They're going to lack peptidoglycan cell walls, and they're going to be facultative or obligate anaerobes. They're believed to be present in small self-replicating life forms. So some other low G+C bacilli and cocci, you have what's called bacillus, that's aerobic and facultative anaerobic. They're going to have endospore forming abilities. They're found in association with the soil and rotting vegetation. They're going to be beneficial in agriculture and in production of antibiotics, and others cause anthrax and food poisoning. And then the last one, you might hear about this one quite a bit in the news or with the FDA, the listeria outbreaks. This is one that's commonly going to be found in refrigerators and around food. So you often hear about foods being recalled because of listeria. Now, listeria is going to be non-endospore forming. They continue to reproduce under refrigeration, so that's why we often find them in foods that are refrigerated because they can still reproduce. It does not slow their metabolism down. They survive inside phagocytic white blood cells, and so that's how they can make you really sick. The white blood cells do not destroy them. They contaminate things like milk and meat products and can cause the bacteremia. That's bacteria in the blood, and they can cause meningitis, so they could be very, very dangerous. Some more low G+C gram-positive bacteria that we're going to have are lactobacillus. These are non-endospore forming, and they're going to be used in the production of yogurt, buttermilk, pickles, sauerkraut, and sourdough bread. They're normally going to be found in your mouth, they're found in your gastrointestinal tract, and they're even found in the vagina. They rarely cause disease. The next you see is going to be streptococcus and enterococcus. So with these two, they're typically non-motile, which means they can't move. They are short bacilli arranged in pairs or short chains. Most are going to be parasites in humans and other animals. So several forms are going to be pathogenic. They'll cause things like strep throat, scarlet fever, rheumatic fever, bacterial pneumonia, and things like that. It also is known to infect wounds. It is known to cause blood infections and infections inside the intestines. And the last one here is going to be Staphylococcus. This cocci is going to be arranged in grape-like clusters or groups, clumps. They're going to be found in association with humans and other animals living on the skin and in the nasal passageways. Some are going to be pathogenic, and they can cause things like pneumonia, toxic shock syndrome, and other infections. So if you get like a staph infection in a cut or in a wound, they would be caused by that type. Now, high gram or guanine plus cytosine gram-positive bacteria is going to refer to bacteria with ratios of guanine to cytosine base pairs above 50% in the genome of the bacteria. So there are going to be three main groups. You're going to have your Corneobacterium. These are irregularly shaped bacilli. They're called pleomorphic. They're enlarged or kind of club-shaped ends on the edges. They're aerobes and facultative anaerobes. They contain metachromatic granules inside of them and inclusions that contain phosphate. So these are going to be like little particles that you can see inside of them, and they contain phosphate inside those particles or inside those little granules. They reproduce by the snapping division, and they're going to be best known for their species of like C. diphtheria. That's going to cause diphtheria in animals, in humans. The next one is Mycobacterium. It's aerobic bacilli, slightly curved to straight rods. They're going to have unique resistant cell walls that contain waxy mycelic acids. This is going to cause them to be resistant to staining and decolorizing. They have to use things like acid-fast stains in order to identify them and see them. They're very slow growing, and they're going to be more common in like soil and water environments. And these are actually the ones that can cause things like tuberculosis and leprosy. The third type is going to be the actinomycetes. These are going to resemble fungi, and they produce spores and form filaments. Now you're going to have three different genera of these. You have the actinomyces, which are going to be facultative carbonic, and that means carbon dioxide filaments. Normally these are going to be found in the human mouth and throat. You have nocardia. These are going to be aerobic organisms that are going to be found in soil and water environments. They tend to form mats of branching thread-like strands or hyphae on the surface of agar. And so if you're looking at them on a petri dish, they kind of look like almost like a type of fungus forming. Now they're going to be useful in degradation of pollutants, and some are going to be pathogens that can cause lesions on the skin. Next, you have your streptomyces. These are going to be filamentous organisms. They're commonly found in things like soil and rotting vegetation. They're primary source for antimicrobial, anti-cancer, and antifungal drugs. So this is going to make up your tetracyclines, your streptomycin, your migranstatin, your amphotericins B. And so they're going to cause a earthy type smell if you smell them, and release geosmin. Now we're moving to gram-negative proteobacteria. This is going to constitute the largest and most diverse group of bacteria. All bacteria are going to be gram-negative and share several nucleoid sequences. They're distinguished by minor differences in rRNA sequences. So we're going to have 5 distinct classes that are designated with the first 5 letters of the Greek alphabet. So the first one is alpha proteobacteria. These are aerobes that are capable of growing at very low nutrient levels. They contain unusual attachment extensions called prosthetica, and they're going to be used for attachments and to increase surface area for nutrient absorption. Several different genera are going to be nitrogen fixers like Azospirillum. These are going to be important in agriculture. They reduce atmospheric nitrogen to ammonia gas, and that's going to be important by providing nitrogen in a usable form for plants. So they also are going to help stimulate root hair production. Remember, root hairs are going to be what are able to absorb things into the plant, things like water, things like different nutrients that are coming from the soil, and even oxygen that it needs. And this will increase root surface area for that nutrient uptake. Okay, rhizobium is going to be your next one. That's going to be important in agriculture as well. It also reduces nitrogen gas into ammonia. That is going to be the same importance because it makes it usable for plants. So it's able to take in its nitrogen this way. It develops symbiotic relationships with roots of things like legumes, peas, soybeans, clovers, alfalfa, beans. All of those different things are legumes. And they'll produce nodules that make ammonia available for that plant during growth. And then you have your rhodopseudomonias. These are going to be important in agriculture because they reduce that nitrogen into ammonia again. And that is also, like we've just said, for the past two, going to be important because it makes that nitrogen a usable form for those plants. This is also going to be known, though, to help reduce hydrogen into hydrogen gas, and that can be used for a clean burning fuel. Next, we have our nitrifying bacteria. And so here we have Nitrobacter. That's going to be important in agriculture and the environment. It converts and reduces ammonia, ammonium ions into nitrates. It is a process that's known as nitrification. And so that nitrate moves better through the soil and it's going to be more available to plants. Next, we have our purple non-sulfur phototrophs. They're going to grow in upper layers of mud in lake and pond bottoms. They may be rods, they could be curved rods, or they could be spirals. They're going to harvest light using bacteriochlorophylls. They do not generate oxygen during their photosynthesis, though. They could be pathogenic alpha bacteria. That's going to be things like Rickettsia, and that's going to be tiny, non-motile, aerobic rod-shaped organisms that are going to grow only within the cells of a mammalian host. So it has to be in a mammal. These are going to be transmitted from one host to another by biting insects such as lice or ticks. They're going to be known to cause things like Rocky Mountain spotted fever and typhus. And the last one on here is going to be Brucella. This is going to be a coccobacillus. It causes brucellosis. That's going to be characterized by spontaneous abortions or sterility. Humans, it will cause things like chills, sweating, fatigue, and fever. It's transmitted to humans by arthropod vectors or by other means. Some other alpha bacteria. These are going to be the Acetobacter or the Gluconobacter. They're going to be important in industry and environment because they're going to be used to synthesize acetic acid, or what we know as vinegar. You have your Caulobacter. These are going to be common prosthetic rods, and they're going to be microbes here that are going to inhibit nutrient-poor seawater and freshwater. And then you have Agrobacterium that's going to infect plants to form tumors known as galls. And so this is going to occur as a result of gene transfer to plant cells. Galls are going to produce nutrients for bacteria. Now we're going into our second class of proteobacteria. We've done alpha, now we're doing the beta. So beta protobacteria are going to live in habitats with low levels of nutrients, and they have several different genres as well. They're going to be pathogenic beta proteobacteria like Neisseria. They're going to be diplococci, so you'll have two cocci together, and inhibit mucous membranes of animals. They cause diseases such as gonorrhea, meningitis, and pelvic inflammatory disease. Next, we have Bordetella. This is going to cause pertussis, or whooping cough. And lastly on here, we have Burkholderia. That's going to recycle organic compounds in nature, and it's going to help to colonize the lungs of cystic fibrosis patients. Non-pathogenic beta protobacteria are going to be like Theobacillus. That's going to be a colorless chemoautotrope. It's important in converting sulfur-containing compounds such as elemental sulfur, sulfides, and sulfites into sulfates, which is SO4. These sulfates are going to be used by plants to synthesize amino acids, and they're going to use those to build proteins. Sulfates are going to be very important for that, for plants. So they're used in commercial mining operations as well. They're able to oxidize iron and inorganic sulfur compounds, and they can produce sulfuric acid as a byproduct. That's going to help to dissolve minerals such as copper and uranium from low-grade ores and make the recovery of these materials cost-effective. They're also going to be used to remove sulfur from coal, and that's going to reduce the potential for air pollution when we burn coal. Next type, we're going to have zoogloia, and that's going to be used in sewage treatment plants, so it forms flocks. Flocks are going to be slimy, tangled masses of organic matter, and flocks are going to be compact and help to assist in purification processes. And lastly, we have the Spherotilus, and these are going to block sewage treatment pipes. They're going to form flocks as well, which are the slimy, tangled masses of organic matter. And the flocks are going to be loose to impede proper flow of waste through that plant. So they can cause a lot of problems. Next, we're going to go into the third class. Now we're in gamma. So gamma proteobacteria. This is going to be the largest and most diverse class of proteobacteria, and there's going to be several different genera here as well. We have purple sulfur bacteria, which are obligate anaerobes. They're going to oxidize hydrogen sulfide into sulfur and deposit as internal granules. They're going to be found in sulfur-rich environments such as lakes, bogs, and oceans. And they're also going to be intracellular pathogens. Next, we have Legionella. That's going to be free-living microbes that are going to be found in water or damp soils. Often they're going to colonize water supply lines. They grow and reproduce within freshwater amoebas. They're going to make them difficult or hard to eradicate. They're going to thrive in white blood cells. They cause things like Legionellus or Legionnaire's disease, which produce high fevers and pneumonia. We have Coxella, that's going to thrive in white blood cells. They're going to grow best in low pHs, and they're going to cause Q fever. That's going to be mild flu-like symptoms. They have severe headaches, vomiting, and diarrhea. They're going to have methane oxidizers. These are going to utilize methane as carbon and energy sources. It's important because they can digest most of methane that's going to be produced by methanogens before they adversely affect the world's climate. So they inhibit anaerobic environments. You also have glycolytic facultative anaerobes. These are going to be the largest group of gamma proteobacteria. They are going to be facultative anaerobic rods. They catabolize carbohydrates by glycolysis. That means they break them down through glycolysis and the pentose phosphate pathway. You'll have things like Escherichia coli, or we refer to it as E. coli, and that's going to be the most studied organism. This is going to form as part of a normal flora within the large intestine or colon of humans and other animals. This is actually a very important bacteria. Lots of people talk about having E. coli poisoning or food poisoning from E. coli and getting sick, but you do need this in your lower digestive tract because it does produce things like vitamin K that you're able to use. Some strains are going to be serious pathogens, though, and they are responsible, like we said, for food recalls and sometimes death. And that's going to be the O157 and the H7s. So 3 important human applications of E. coli. We have water testing. So we use E. coli as indicators of fecal contamination in water. We use it in research. We research different strains and do genetic research with it. And we can, thirdly, use it as a recombinant protein production. So we often use E. coli to produce proteins that are found in humans. And we make large amounts of these in order for the pharmaceutical companies. So things like insulin and human growth hormone and erythropoietin or EPO are often going to be made in E. coli in large amounts. And then we're able to use them to treat things such as diabetes. Pseudomonads are going to be aerobic flagellated bacilli. They're commonly going to be found in soil and water environments. So you have your Pseudomonas They are going to cause urinary tract infections and swimmer's ear. It's used in environmental cleanups because they can utilize diverse assortments of organic compounds as carbon and energy sources. They're used in cleaning up oil spills during petroleum transport. They're used to clean up insecticides, herbicides, and gasoline that were inadvertently introduced to an environment. Next, you're going to have your Azobacter and Azomonas. These are going to be soil-dwelling, non-pathogenic nitrogen fixers, and they're not going to be associated with plant roots, though. So these are the type that don't really help with plants, so they don't help with agriculture. Now, our fourth one in this class is going to be the Delta Proteobacteria. So we have Desulfovibrio. This is going to be responsible for reducing elemental sulfur and sulfur-containing compounds, resulting in the formation of hydrogen sulfide gas. So it's going to inhabit the sediment of polluted streams and sewage treatment lagoons. Bdellovibrio is going to attack and kill other gram-negative bacteria. They're going to live within periplasmic space, that's going to be space between cell walls and cell membranes, and they're going to kill the host by disrupting cell membrane and inhibiting things like DNA, RNA, and protein synthesis. And then our last type here, which is the Myxobacteria, is going to be aerobic soil-dwelling bacteria that's going to live in temperate regions within decaying plant material or animal dung. And this is our fifth class. We're going to have the Epsilon Proteobacteria. They're going to exist as rods, vibrios, and spirilla. First one here, we have Campylobacter. That's going to be blood poisoning and inflammation of the intestinal tract. And then we have Heliobacter, and this is going to be responsible for causing things such as ulcers in the intestinal tract. Next, we have chlamydias. So chlamydia is going to be small coccus-shaped microorganisms that lack peptidoglycan in their cell walls. They're going to reproduce only inside of host cells such as mammals, birds, and some invertebrates. They do not require vectors for transmission, and they're going to be transmitted to humans through interpersonal contact, or they may become airborne or infect via the respiratory tract. They're responsible for things like trachoma, an infection of the eye that's often resulting in blindness, pneumonia, and the sexually transmitted urethritis, and that's going to be similar to gonorrhea. So most commonly, it's going to be reported sexually as a sexually transmitted disease or STD. Next, we have the spirochetes. These are going to be helical motile bacteria that have flexible cell walls. They're going to contain periplasmic flagella. These are going to be flagella that's located within their periplasmic space that's providing motility for that organism. Very thin and poorly visible. So they exist as free-living aquatic forms. They can be parasitic, they can be pathogenic to humans, and we're going to see three types here. So we have tryponemia, that's going to cause agents of syphilis. We have Borrelia, that's going to be the causative agent of relapsing fever from Lyme's disease. And we have Leptospirea, and that's going to be the causative agent of infections resulting in fevers such as jaundice and meningitis. And lastly, today we're going to talk about bacteroids. These are going to be obligate anaerobic rods, and they're going to inhabit the digestive tracts of humans and animals. They're going to help to assist in catabolic digestion of things like cellulose and complex carbohydrates the body normally can't digest on its own. And so some species of these, though, can cause infections. Then we have our cytophagia. That's going to be aquatic, gliding, rod-shaped aerobes. They are going to degrade complex polysaccharides that are going to be found in things such as wood and raw sewage. So that's going to end us with our prokaryotes. We will pick back up with our eukaryotes in our next video. So see you with the next video.